Hey there, 7th graders. Today's lesson is 2.2 microbiomes. Your learning targets for today. I can explain the importance of good bacteria and the human microbiome. So today we're going to learn all about the human microbiome, which is basically all the microscopic organisms that live in and on our body. And you guys will also be introduced to patient 23, which is a case study we're going to be talking about here in class. A case study is kind of when we look at someone's medical records and try to figure out what's going on with them and what we can learn from that information or that data. Okay, moving on to activity 1, which is your notebook. You guys have key concept 5 and 6 to add into your key concepts page. So they will go right here. The human microbiome contains approximately 100 trillion microorganisms. So that's not just bacteria, but most of that is bacteria. Most of these are bacteria. And then key concept 6, the human body provides an environment, which in the environment it includes food and space for bacteria to survive. You guys also need to add bacteria and microbiome to your vocab. So again, that would look a little bit something like this, where we've got bacteria right here, tiny organisms that are made of one single cell. And they obviously belong into that kingdom bacteria. And microbiome is all the microorganisms that live in a particular environment. So we could look at the microbiome on our desk. We could look at the microbiome on our phone, which some of you guys are doing that in class tomorrow. Okay, such as the human body. All right, so that's your guys' notebook ad for today. Moving on to our warm-up, which is also part of your bell ringer. So last week you guys got a picture of a petri dish, and you guys were asked to see if there was any evidence of microorganisms growing on it. And then 9 days later, you guys have this picture here. So using your draw tool, you guys can circle yes or no. Can you see any evidence of microorganisms? And then this is going to be similar to what you guys maybe start seeing. You guys will probably have a little bit more because yours will be a little bit denser in your plates, but this will be similar to what you guys will see in lab as well. And then question 2, describe what you observe in the petri dish. So give me a really good description. Talk about the different types of things you see. So this is really good practice again for that lab doing your daily observations. All right, remember that as you go through these videos, you can pause them as you need to complete each section I just explained. So moving on to activity 3, you guys have a little movie to watch. So you guys can right click and open that link. It's going to take you into YouTube. And then you guys have this video called The Bill. So it kind of relates back to that Joe Naismith campaign ad that I showed you guys. It talks a little bit about what his stance is on our next part that we're going to talk about, which is fecal transplant, and of course the science side of it. So go ahead and watch the bill. You guys can pause this video, and then when you're done watching that, come on back. All right, moving on to activity 4. Here is patient number 3, an introduction. So at the Microbiome Research Institute, we collect data from many patients that have undergone a fecal transplant procedure. Basically, feces or a fecal, the word fecal, means poop. And I know that sounds gross, but this is an actual procedure that we can do in labs to kind of correct people's gut microbiome to make them feel better. We collect information about these patients so we can more clearly understand what is going on in their gut microbiomes before and after the procedure. This practice of carefully collecting data about one patient is called a case study. I'm sending you a case study about patient 23, a 64-year-old man. We collected data about this patient's gut microbiome over 9 weeks while he was undergoing a fecal transplant procedure. At the end of the 9 weeks, he was cured of a potentially deadly infection. So that's going to be that C. difficile that we talked about in the card sort the other day. How did the fecal transplant procedure cure this patient of a very harmful infection? Please analyze the information collected about patient 23's gut microbiome to help us answer this question as we build our argument against the senator. So again, you guys saw kind of what the senator had to say in the video you just watched. Okay, moving on, here's kind of what we're going to be doing with his pie charts. So for our data for patient 23, we're going to be looking at some pie charts. So we're going to annotate the case study pie charts below with your comments and questions. Then discuss the following questions. So you guys don't have to discuss your questions with your partners. You guys are working from home. But think about these answers to these questions. What is the same about the patient's gut microbiome data from week 1 to week 3, and what is different? Those would be good things for you to annotate as well. When you are finishing discussing with a partner, answer the questions about the patient at the bottom of the screen. All right, so let's take a look right here. We've got data from week 1, so we can see the pie chart. We can look and read and see, all right, this light blue color, which is almost half of his gut microbiome, is this bacilli bacteria. We got B. fragilis, which is this darker blue. We got this kind of bacteria right here, actinobacteria, E. coli bacteria, and just other types of bacteria kind of grouped into that one pie. That was on week 1. Okay, the patient is feeling normal on week 1. Week 2, you guys note the nice little icon of the patient visiting King John there and ralphing a little bit. Okay, you guys can see his doctor notes, patient reports vomiting and diarrhea and a fever. So take a look at the pie chart here and observe what's different. What you guys will do is you need to put in at least 3 annotations. So you guys, again, can focus on things that are the same, things that are different, or other things you guys might notice about the information you see here, maybe how the patient feels, whatever the case. So you guys will type what you notice in these 3 boxes, and then you guys can use your draw tool to connect. So maybe I type here that, remember when you use your draw tool to click back on your cursor, maybe I type here that the patient is sick on week 3, okay? And then I take my draw tool and I'm gonna kind of highlight that right here, okay? So now I kind of got a little cue in to show me how that patient's feeling between week 1 and week 3. Okay, again, anytime you guys use your draw tool, make sure you guys click back on your cursor, otherwise it just won't work for you guys. So complete at least annotation 2 and 3. You guys can use the same as I used for 1, or you guys can come up with your own if you see something big you want to annotate. Okay, record your ideas below about why you think patient 23 feels normal during week 1 but sick during week 3. And the evidence that supports my ideas is, so go back to this pie chart and give me some specific evidence, okay? I should see some data in that question number 2. Okay, activity 5, we're gonna learn a little bit about the gut microbiome. So go ahead, right click and open this link up. It's gonna take you guys into Amplify, and you guys get to read this article. Okay, remember that my bargain with you guys is you don't have to highlight and annotate this article as long as I can trust and assume that you guys are doing a good job reading it. In this case, you guys have some questions that you need to answer. So if you're not doing a good job reading it, you're just not going to do a good job answering those questions. And if that's the case, if I feel you just didn't read it, then you guys, obviously, we will be back to highlighting and annotating. All right, remember you can also listen to the article or listen while you read. If you guys click the speaker and then hit play. It's going to buffer a second.
Microbiome.
Okay, so that's an option for you guys as well. So go ahead, pause this video, take a couple minutes to read the gut, a human microbiome, and then move on to these questions. While you guys read, you can look for these. What do bacteria do in a healthy gut microbiome? So why are bacteria important for a healthy gut microbiome? What is one type of bacteria found in a healthy gut microbiome? It talks about several types in there. What is a type of harmful bacteria found in the human gut microbiome? So what's a bad one that we would find in there? And what do harmful bacteria do in the gut microbiome? Finally, use what you learned in the article to explain why patient 23 feels sick. Okay, so again, you guys saw the information about patient 23 in that introductory pie charts on the case study. But now you guys have a little bit more scientific background from the article. So what did you learn in that article that's going to kind of give you guys a little bit better description as to why patient 23 is feeling sick in week 3? Okay, finally, complete your wrap-up circle. Do you have any questions, yes or no? Remember, this is required. You do need to do this. If you have any questions, type them right here. If you don't, no big deal. And then circle 4, 3, 2, 1. How do you think you did on this assignment? You are all done. Good work. Now go ahead and hand this in. Bye.